She received her PhD from the Joint Doctoral Program at University of California, San Diego and San Diego State University. And to date, her research has focused on a number of topics, including things such as sleep, PTSD, health psychology, and what she'll be presenting on today, the social rhythm model of mood disorders. To date, her research has been awarded numerous funding awards from various agencies, including the Department of Defense, the VA system, more locally, the Institute for Mental Health Research. And in addition to giving her money to do her research, they've also given her a number of awards for the research that she has put out to date from many of these same agencies. Dr. Haynes, look forward to hearing your talk today.
Thanks so much. What a nice introduction. So today I'm going to, several of you might have heard me speak in the past about social rhythm models and a therapy we've been developing called cognitive behavioral social rhythm therapy, which is strongly based on the model. But today I've chosen to take a step back and just explain like what the heck are social rhythms, why they're important, and kind of what they have to do with circadian rhythms and sleep. And then sort of progressing you through my research in terms of how we've explored this construct and where we're going. Okay, so my hope is that this is a part informative, like clinically based talk, as well as sort of kind of interspersed with some research findings out there. Alrighty. Okay, so there are four main questions that I'm really hoping to get to in this talk, with the first one being, what are circadian rhythms? I mean, we all know, what a circadian rhythm kind of is, but the more we ask about this definition and we learn about things that drive circadian rhythms, the more we actually question what is a circadian rhythm. We're going to talk about, I'm going to do some defining of something called, this great word called Zeitgebers, and then also talk about masking effects, because, as much as we think we know these are, I hear constantly terminology, people getting confused in the way they use these terms. The next thing I want to address is there such a thing as a non-photoic Zeitgeber? And then what are social rhythms? Are social rhythms even really rhythms in the circadian sense? And that's a big kind of debate out there. The third thing I want to talk about is what role do social rhythms play in depression and anxiety? And then finally, how can they be applied to treatments for depression as well as for anxiety too? So hopefully we get a chance to address these today. Okay, circadian rhythms 101 slide, and this may look familiar to some people in the audience. Circadian rhythms are cycles that they're internal cycles that are internally generated that occur generally on a 24-hour cycle. Okay, so I like this slide here because it kind of talks about, it divides, first of all, one of their major rhythms, which has to do with behavior and sleep. So one of the things we know in humans is that generally, and obviously not all humans, like shift workers might differ, but generally between about 11 o'clock to about 6 a.m. we're at the lowest behavioral cycle in terms of their circadian rhythm. Okay, and there's a number of things that are happening at that time, other rhythms that are occurring. For instance, melatonin secretion starts. Okay, we also know there's very little light outside if we're corresponding with the darkness. We also know that body temperature starts being very high about 7 p.m. and is dropping throughout this time till we get to the lowest body temperature, and it starts to rise about the time generally we often wake up, right? Because this is what behavior is very low at that time. There are other body circadian rhythms too. I just want to point out melatonin is one of the most powerful things we talk about because it's such a nice marker of sleep and behavior, as well as body temperature. But there are other things like blood pressure that's on a circadian rhythm, testosterone secretion, alertness, coordination. All of these occur on 24-hour cycles. Okay? When we talk about sleep and wake, we generally don't talk about sleep and wake as a circadian rhythm, but more as a homeostatic process, although it does occur within a 24-hour cycle too. Okay? So that's just sort of my general overview of what is a circadian rhythm. Okay? What coordinates our circadian rhythms? What gets it going? Well, generally, we think, okay, circadian rhythm clock. Where's the clock? The clock is in the suprachiasmatic nucleus of the hypothalamus. But interestingly, when we look at these pathways, one of the biggest things I like pointing out here is that there are actually very few direct pathways between the suprachiasmatic nucleus and these rhythms exactly, like the thermoregulation or sleep. A lot of it is a secondary pathway. It goes through the dorsomediolateral nucleus of the hypothalamus. That's our secondary input. And then from there, things get relayed to, for instance, the medial preoptic nucleus, and that's what regulates like our temperature, thermoregulation. The DMH also has projections to the ventrolateral preoptic nucleus, which is where we look at light for sleep, and that tends to impact our sleep. We have the lateral hypothalamus, and this is where we think about orexin and melanin, where wakefulness and feeding come into play. Now, it's interesting because we have these appetite hormones and appetite suppression hormones that actually don't even necessarily work directly through the suprachiasmatic nucleus. They work through the ventromedial nucleus, the arcuate nucleus. So I think it's important to look at the system is much more complex than just having one master clock. There are a number of different, there may be one oscillator, but there are potentially two, right? And then a number of different systems that kind of go in. Okay. Having a hard time, I usually do return. Let's see if I can go this way. Nope. Okay. All right, I'm going to advance. There we go. Okay, what is a Zeitgeber? Okay, so Zeitgeber is German for time giver. Zeit is time, and then Geber is giver in German. And generally, a Zeitgeber is something we call an entraining agent. Okay, so it's the thing that's in our external world that synchronizes our internal clock to the external cues. So it's a really important structure that helps us regulate so that we can live, so that we naturally want to kind of go to sleep when the sun goes down and kind of wake up when the sun comes up. So that sort of leads to the next, my goodness, there has to be a better way. So it's the next issue of light being our most powerful Zeitgeber, and I'll talk about that in a minute. When I use the term phase shift, it really refers to resetting the clock due to an entraining agent, okay? So let's say you fly, let's say you do shift work or you have jet lag, okay? And so this would be when you're going through something called, you experience something called a phase shift because, for instance, you know, the majority of light that you're getting through the optic inputs, right, through the eyes is going to be occurring when, if you're working graveyard, at night. And then if you sleep during the day, you're going to be getting a lot of darkness. So melatonin suppression hopefully adjusts appropriately. So you're shifting your phase from a typical like day cycle to more of a night cycle. Okay? And the same thing occurs with jet lag. And I think all of us have experienced jet lag intrinsically and kind of know what a phase shift feels like when we fly to Australia or we fly east or then we try to come back west. We kind of see how our clock adjusts, and it typically takes a little while. Okay, now I think one of the most important things kind of getting out of this is that an entraining agent, something that actually sets our clock, it tells us when the clock, you know, it literally sets our clock if you think of it like a clock, is very different than something called a masking behavior. A masking behavior is when the circadian rhythm is modified by an external influence. Okay, so I have the example here would be gastrointestinal functioning. Okay, so for instance, when we eat, our gastric acid rises, right? So to a certain extent, food is a masking agent, okay? Now there's also circadian rhythm for GI function and for appetite and suppression and stuff like that, but if we just, you know, we tend to have, 3 meals a day, right? Breakfast, lunch, and dinner, and if we looked at that, we'd say, oh my gosh, there's this crazy rhythm that's occurring, gastric acid is rising, morning, lunch, and dinner, but no, in actuality it isn't. Food is just masking that, okay? The same thing with light on melatonin. Okay, so if we get a lot of light in the evening, and it's just crazy because this person's melatonin doesn't really kick in until really late. And the reason being is because our light is suppressing it. As a reminder, melatonin, right, tends to release in the evening when the sun goes down. Okay? So light, as I've already kind of talked about, is our dominant Zeitgeber. There are, I would say, you know, compared to the power of anything else, light is what keeps us timed with our external world. Okay, it was originally thought that what was really necessary was outdoor bright light. That was what was necessary for entrainment. anything over like 1,000 lux, for instance. But now we know that humans are actually pretty sensitive to even low levels of light. I qualify that because there are two things that we need to know. For instance, when. So low levels of light in the middle of the day probably don't mean as much as low levels of light that occur, for instance, at midnight. And we call that in circadian rhythm research the phase response curve, when we're most sensitive to the effects of light on our melatonin. Okay? And then there's also a dose response curve with light as well. For instance, how much? Okay, so there has been some suggestion, although I don't think it's really strong, that even levels of light at like 100 lux, so for instance, I would say the lights in here, I would say probably are like 60, 70 lux. Counter me if you think I'm wrong. I'm trying to figure it out, 60, 70. And if you think about it, in the middle of the night, when, for instance, our elderly patients are getting up and getting out of bed, and they turn on the lights because they don't want to fall, that could potentially have an effect on what? Melatonin, right? Melatonin that's being secreted. So, especially in the middle of the night. So I kind of want you to think about like kind of levels of light, but in the middle of the day, when they're getting a lot of sunlight, probably not going to make a difference at all. Okay? So I do want to say that the sensitivity of light increases with the prolonged exposure to dim light. Why is that important? Okay, so what do people with depression do? Do they get out? Do they, kind of play in the sun? Generally not, right? Generally there's probably a lot more isolated behavior patterns. And so if you're not getting a lot of exposure to light, smaller amount of lights are going to matter. Our system adjusts. Okay? Why is there, here I am talking about light, outdoor light, bright light. Why is there this picture of blue light? Well, we found that our system is especially sensitive to blue light in terms of the power of entrainment. That's a relatively recent, and the reason is because of these really fun cells. And I don't know if any of you have ever Googled a cell, like a picture of a cell, but it's amazing the number of people that get tattoos of neurons on their arms that they really like. And so this person said something like, I chose this tattoo because, photosensitive retinal ganglion cells are just really cool. You don't need to see to get them. It's just light. They're in the back of our near the nerve, and you don't have to have sight, but as long as you perceive light, these cells produce melanopsin, which then trigger the cascade of our circadian rhythms. So these are the cells responsible for our perception of blue light, which then lead us to entrain into circadian rhythms. Okay. So there's these really, I would say, landmark experiments by Ashoff and his colleague, and I don't know if any of you learned about them in the past. Maybe I'm dredging up some Psychology 101 here, but they put people in caves for an extended period of time, and it wasn't, and there was a number of variations by which people went to the caves. But the goal was being, if we can isolate people sort of from light in general, although, they don't make people sit around in dark caves, so that was one of the major problems with the experiments. But bright light for an extended period of time, 30 days, even more, and if we can isolate people from sort of social stimulus, what happens to their circadian rhythms? And what these experiments showed is that in the absence of strong, powerful light, in the absence of cues as to their social, like what's happening in the social world, humans free run to about 24 hours. By free run, that means they just kind of get into this pattern of sleeping and eating and waking, and their whole period, and it starts all over again in about 24 hours. And that's pretty cool if you think about it, because that's consistent with the sun, right? That's how we work. So it's pretty amazing that, to a certain extent, I think you can almost get religious on this, it's pretty amazing that our bodies are set up, and most things on Earth are set up to run anywhere from 23 to 25 hours. And how do we study this now? Because we really don't put people in caves anymore. Well, there are two methodologies that sleep researchers use. One is called constant routine, and this is a methodology by which patients are pretty much told to stay in bed, and they can't move really, because moving is masking, right? Because when you move, your blood pressure goes up, your thermoregulation goes up. They feed people intravenously, you don't eat, or they don't eat at all, and it's for an extended period of time, probably not, I think probably like 7 days is about average. Very intensive, very few social stimulus, and from that they can get to the same type of of almost a free running pattern where they're really not timed in with the light or the dark anymore, but you can see that 24-hour period. There's also this protocol called forced desynchrony, which I think is pretty much a landmark. And what they do is they put people into, and I'm kind of explaining it very roughly with the goal of just getting a general idea. There are definitely more specifications by which you can kind of understand these protocols. But Sleisler came up with this, and you put people into a sort of, again, a place where you're for an extended period of days, like, and they go into sort of a containment where they're really not given any sort of social cues of what day it is or what time it is, and they're forced to live on a sleep-wake schedule that makes them break apart from their circadian rhythm. Okay, so you can see that 24-hour circadian rhythm with a sleep-wake pattern, for instance, that's 6 hours, 6 hours sleep, 6 hours awake. I don't think they go to that extreme. Dick, do you know? I don't know off the top of my head, about 6 or maybe more, but that's sort of the beauty of the free run is when you can get sleep, because sleep is a masking, because when you sleep, what happens to your temperature? It goes down, right? When you can break the sleep-wake cycle from the circadian rhythm, and then you can look at the circadian rhythm by itself. Okay, so what does this all have to do with social stuff, like in habitual behaviors and social Zeitgebers? And you'll find in my research I talk more and more about habitual behaviors, because there really is a question, is this even a social, if we know that light is super powerful, then it is, and we certainly know that. There's absolutely no denying that ambient illumination exposure is the most powerful Zeitgebers. Is there really even such a thing as a social Zeitgeber? Okay, so our habitual behaviors really, can they really time our body? Can they really make our circadian rhythm stick? Because, and how do you ask that question? Have people socialize in the dark? That doesn't really work, right? So you can see how correlated the light and the socialization are in our world. The bigger question, and I think a really interesting question, has to do with that masking part. Do habitual behaviors just modify our exposure to ambient light? So for instance, if I'm going to go out and socialize, it's more likely that I'm probably going to get more light in my world. Okay? And then finally, this is sort of the third question, is can habitual behaviors or social rhythms modify the circadian system response to light? So if I'm pretty inactive as a person, does that mean that there might be an interaction with how well my circadian system actually perceives the light and uses it to time me? Okay, so I just want to say, in relation to that first question, is there even such a thing as non-photoic Zeitgebers? I would say there is a little bit of evidence for this in animals. And the best evidence comes from the food entrainment literature. And what they find is that when animals are based on a restricted feeding schedule, they do get an anticipatory increase in activity or body or corticosteroid release right before mealtime. If any of you have ever forgotten to feed your cat, and you walk in, right? You know what I'm talking about. You see that, come on, meow, it's time to feed me, right? And they do see this in certain periods that are there, 23 to 28-hour periods. Okay. With daily competing light and feeding stimuli, when they have both, they're going to entrain to the light-dark cycle. That's still real powerful. But they find, and they do find, and this kind of goes back to that 2-oscillator model, remember, that appetite, that lesions to the suprachiasmatic nucleus do not block food entrainment. So we do see that food, in particular, may operate on sort of a separate system. And if you think about it, food is a habitual behavior. It would be considered a social Zeitgeber too. Okay. We also see synchronized free running, if you think about social in the way most of us think about it, which is being social, being with people, the pleasures that we see from it. We do see that when they get to a free running pattern in various rodents, that these rodents tend to synchronize together. Okay, and we've seen this across a number of rodent species, like diurnal palm squirrels, mouse pups, male golden hamsters, octagon dagus. So it's pretty exciting that, and if you live with a partner, you could probably see ways that your social rhythms go hand in hand with that partner's, or if they don't, the problems that can create as well. Okay, but again, it's that kind of gut feeling. But what about on a scientific biological level? Okay, and I just want to say that how do we test this? Well, when we look at arousing stimulus, sometimes they see phase shifts that go hand in hand too, so that's another sign that there's that biological element there. What about humans? Well, right now we have very little, in fact, no evidence that social cues affect our pacemaker whatsoever. Okay? At the same time, it's really hard to eliminate the confound of low light in so many protocols. We just don't socialize. We just don't do a lot of social things or activities in general in the dark, right? How do you really test this? But we do want to say that in bilaterally inoculated subjects, subjects that have no light perception at all because they have no eyes, we do see significant individualist differences, okay, which means sometimes they entrain to a 24-hour rhythm based on habitual behaviors alone, but sometimes patients don't. So we don't know. We don't know why. One of the best bets in human land is that exercise. Okay, we do see that patients that, and it was, again, low light cues, it wasn't binocularly enucleated subjects, but that we see that they may entrain on a phase response curve, that exercise may have an impact on our rhythm. Okay, but a lot more work needs to be done there. So remaining questions, does the type of social contact make a difference? So what about that sufficiently arousing stimulus, for instance? Maybe like when is habitual versus arousing enough? And then how do we standardize this in humans, right? With models, with animal models, it's pretty easy, but in humans, how do you really standardize social input to our systems when it has varying levels of reward? And then here's the other issue: isn't light pretty arousing? Being exposed to light. I mean, I know here in the dark at 4 o'clock, it's pretty easy to kind of take a little siesta in the back, right? But what about light when we go outside? And how do you separate light from arousing stimulus? Isn't that social? And then in what ways do light and non-photoic cues interact? Okay, so I think a lot more work needs to be done in this area. And then why are these individual differences that we see in subjects without eyes? Okay, so this is a model that I really like from Misselberger. And I think this is a model that I've used a lot in terms of kind of understanding the way the way to understand social zeitgebers. So all of these kind of more thin lines here are ones that have not been proven yet, okay? But things that we know that there is sufficient data for. For instance, let me just kind of walk you through it, is that this is the light-dark cycle. It's the sun coming into our eyes. And we know that the more light we get, the more social stimuli we get, the more we're able to see and hear things and smell, like, because we're awake, we're aroused, right? And then that has a major impact on our behavioral state, like what we're choosing to do. Okay, so unless you have some form of extreme sleepiness disease, if you're in a very stimulating environment, you're much more likely to be more active and be more alert. And then ironically, the more active and alert that we are, the more likely we are to actually get exposure to light. Okay, and you can see how that sort of works. We also know that light has an impact on the circadian clock directly. These things that I think need a lot more research is, does behavioral state affect our circadian clock or even moderate it? Okay, is there an interaction effect there? And then does behavioral state affect our circadian clock as well directly? Right? One thing I want to point out, so I think that it's just really important to remember that behavioral state and goes this, that this arrow can go this way. The behavioral state definitely affects our exposure to light, dark, which is important to remember when we think about activity, and even things like behavioral activation, these treatments that we use that are considered pretty important. Okay, so that kind of leads me into how does all of this relate to depression? Well, first of all, I just want to take a step back and look at what we know from the depression literature in terms of some of the chronobiological abnormalities we see there. First of all, depression's been characterized by a disruption of circadian rhythms. And I won't go into specifics there, but there are a number of ways people who've looked at body temperature and cortisol and kind of phase, and we see a lot of differences there. And there's a number of theories that have been developed. So one theory is that maybe people with depression are desynchronized to their sleep-wake cycle is at a different place from their circadian cycle. And some people think it's a phase advance, although it's been really unclear in the literature what is advanced. Is it the sleep that's advanced or the rhythm that's advanced? Some people think that there's a theory called the blunted circadian rhythm theory, which is that people that are depressed, they're just going to They're not going to have, like most of us have these activity patterns where we're really high in the day and really low at night, right? But people that are depressed might have a lot less in the day and a lot more at night. So it's blunted cosine, a blunted wave. Finally, some people have suggested that maybe people with depression are just not as responsive to light or Zeitgebers in general. Or maybe they're hypersensitive to it. So again, we need more research, but we do know that we see on the output side of things, we do see a number of sleep abnormalities that occur in depression. And this is pretty consistent across the literature. In fact, more than any other disorder out there, we have a lot of good data that there's some serious chronobiological abnormalities in depression. And the four that we see, the four hallmark symptoms are poor sleep maintenance, okay, so difficulty maintaining sleep, reduced levels of slow wave sleep, and if you remember slow wave sleep, the function of that is really that restorative sleep, altered distribution and increased activity of REM sleep. So generally, and this kind of, I think 3 and 4 kind of goes together, what we see is that people go into REM much earlier, and they tend to have much more REM, and the REM that they have is much more dense. Okay, so people have looked a lot at that. So it's a pretty interesting phenomena. We also know that people with depression have some abnormalities in the way that they perceive light. Okay, and I say people, but I also want to look at models. So if we look at an animal model, we know that animals that are kept in constant darkness have depressed profiles, including the behavioral and monoamine systems look very similar to what we would see in depression. Okay. Also, seasonal affective disorder, probably something we don't see tons here in Tucson, but up in the Pacific Northwest, right, really frequent. And that's a really good model because what is the motivating hypothesis behind seasonal affective disorder? It's that shortened photo period, the shortened amount of light the light that people get in the winter in our hemisphere. So that would be another sort of idea that maybe there's some light abnormalities going on in depression as well. There's also, I'd say 2 studies out there that I'm aware of, although there may be more at this point. In the last 6 months, I haven't seen a lot, though. But we know that low levels of bright light are associated with increased depressive symptoms. And they monitor that via something called an actigraph, where you can actually get some very nice objective data on the amount of light people are exposed to. We know that PER2 is a gene that's associated with vulnerability to depression, and it's also a period gene. It has to do with circadian rhythms and chronobiology. And then finally, I think this is one of the most interesting studies, is ocular pathology. Okay, and I'm not just talking like glaucoma or things like that, but ocular pathology, even I think they were even looking at non-20/20 vision, contributes significantly to depression. Okay, so interesting things. So again, more data is showing that maybe people with depression are having some form of issues in their ability to perceive light. And when we ask them, hey, does it feel dark in here? Does it feel light in here? We know that those patients that tend to have more depression, this is obviously, the N was much higher, but I kind of graphed it from one of their things, that those patients who said that they're more depressed are more likely to acknowledge experiencing dim light than those patients who are less depressed. All right, behavioral state abnormalities. Okay, another finding that we see in depression quite a bit. Depressed individuals in general engage in a lot less activity than non-depressed individuals. And this is a whole different body of literature and psychology that we know. We know people in depression, though, but when we look at activities, there's a lot of different ways to look at activity, right? One way of activity, a lot of times we think about doing pleasant things. And that's certainly true in the literature, less rewarding or less things that provide control or mastery over your environment. We also know that when we get really literal with the term activity and we just measure motor movement, we know depressed individuals have less of it. They don't move as much. They have less exercise, less physical activity. They generally have smaller social networks, so fewer activities that are social-based. And they also know that they have fewer habitual behaviors. By this, I'm talking about just elements of their routine, like eating, seeing people, walking their dog, exercise, things you might do on an everyday basis. They have fewer of those, and generally that fewer of those are coupled with worse sleep. Okay. Not to skip around, it all comes together, but I want to bring in one more body of literature from the depression area. Okay, so we've talked about light, we've talked about sleep, we've talked about behavioral, now I want to talk about life events. Okay, so life events, very stressful life events. And what we know is that severe life events predict major depressive episodes. Okay, they're one of the best predictors. But we only know that it only occurs in some people, people that are vulnerable. And a whole body of literature has investigated, okay, what makes people vulnerable? Because some people go through life stress and they do great. They don't get depressed. So we know early stress like childhood abuse makes people vulnerable. A lack of a support system, you might have heard of something called the buffering hypothesis. So when people don't have the support system and they go through stress, they're more vulnerable for depression. We know that when they have a cognitive bias, they're more likely just to think like, I suck as a person, or it's all my fault, or it's always going to be this way, or those attributional styles. We know that they're more likely to get depressed. And then there's those genetic and biologic vulnerability factors that we could go into. A whole different thing of that. But what about that chronobiology disturbance? Okay, and that's where the social rhythm theory of depression come in. Okay, so this is the model that we're talking about, the come together model. Okay, so according to this model, a disrupting life events occurs in depression, which then has an impact directly on people's social rhythms, or if we want to call it habitual behaviors or daily routine. Okay, but something gets disrupted, which then negatively impacts how they sleep. So if you think about it, let's just kind of walk through it. A person has an argument with a significant other. They stay up late fighting, right? They decide to sleep in late the next day, which then impacts like the amount of light they're getting and that exposure and how they feel during the day. Okay, or you can kind of go this way, right? Because exposure to light or direct disruption, no one really knows exactly, which then could set off a mood dysregulation in a person that's more vulnerable. A person that's not vulnerable, it kind of bounces back the next day. Okay. So how do we measure these habitual behaviors? Okay, and the major way, and this is the measure that we use in my lab all the time, it's sort of the major construct that we look at, is something called the social rhythm metric. And I just kind of wanted to point out the activities that are considered the habitual activities, things like getting out of bed, having contact with another person, starting work or housework or volunteer or childhood or family care, having dinner. You can see that the majority of these activities are not activities we would think of particularly as giving us tons of control or mastery. I mean, people could argue that, right? But it's not like going to the movies or, having a really great date or, something that we would consider like inherently reinforcing. Okay, these are kind of basic habitual behaviors. And why are these ones kind of highlighted in red? Because they've actually found that these 6 are the most important. Okay, so we've done analyses and factor analyses and taken them down. Getting out of bed, having contact with the person first thing in the morning, starting work, having dinner, and going to bed are the major things that really play a role. And I think it's interesting that there's at least one meal in there, and then our activities are in there. And that there's 2 sleep-related ones in there. Okay. So this is from a study we looked at, and what we found is that in a group of about 74 patients with 50, and I want to say it was half the sample was depressed and half was not depressed or had no history of depression. All patients were between the age of 18 and 65. We found that those individuals who had higher social rhythm scores. Okay, so let me go back and talk about that. Sorry. Social rhythms, and our social rhythm score would be not only the frequency with which they're engaging in the behavior, but the variability by which they're engaging in it. Okay, sorry about that. So if I go back here, so if I had breakfast at like 8 a.m. on one day and then 10 a.m. on the next, that would give me probably that would contribute to a lower score. Okay, that would be highly variable, because what we're looking at really is consistency. Okay, so what we found though is that those patients that have higher social rhythm scores, more consistent daily routines, they better fit to that cosine curve, the amplitude that I was talking about earlier. Okay, they have lower amounts of nighttime light exposure, which is exactly what we'd want to fit to that rhythm. They have an earlier bedtime in the evening and an earlier wake time in the morning. And I think the reason I really want to bring that up is because if you think about it, has anyone ever gone backpacking or lived on a farm or anything like that? This is the type of lifestyle you have where you're not relying upon electricity as much. Electricity that makes us stay up later, get up earlier. With something like this, you're much more consistent with the sun, right? So it fits our construct nicely. So these are scores between the normal control and the depressed, okay? And what we found is that normal controls in general, it fits nicely, have much more consistent routines than depressed patients. And then we also found, and I want to say there are mean differences, but where we found significant differences, we also found that normal controls also have a lot more activities with other people actively involved in their routine than depressed people. Whereas the majority of their activities, I mean, in general, depressed people have fewer activities in general, right? That's the activity level index. But they also have fewer with other people that's passively involved, but they have more activities alone. Okay, so this active engagement in others might be important, and I hope that as I kind of mentioned that you kind of think back to those little picture of two rats sort of snuggling together, right? And they synchronize and phase shift together. There might be that circadian rhythm element. Okay. So goodness of fit to the cosine curve is one way we look at circadian rhythms, like how well are people fitting to that nice amplitude, that cosine curve that tells about the circadian rhythm. And what we found is that so if you look at the purple, this is depressed, is that in general, depressed people, that the more they fit to the cosine curve, the more activities they have, whereas there's kind of a lesser fit that way. But I think what's really interesting is the variability in the depression, right? There's not a lot of relationship there, whereas if we look at individuals who aren't depressed, the normal controls, we can see that those who don't fit to the cosine curve kind of rely on activities more, right? It kind of goes the opposite direction. Okay, so how do I make sense of this data? Is that I think people kind of who are normal controls or people who don't have that vulnerability to depression, I mean, to a certain extent, there's only a couple outliers here. They kind of center around a nice sort of fit. And some people might do better with lower activity levels or higher activity levels. But in general, a pretty good fit to the cosine curve has to do with a lot of activities, more amounts of activities, because it's only like 12 to 14 difference. Okay, let's look at WASO. So this stands for wake time after sleep onset. This is a sleep index. And what we see here is that individuals who aren't depressed, right, who have high levels of activity in general, have lower amounts of time awake after sleep onset. So they have less sleep continuity. They have less sleep continuity issues. And then, kind of, let's say, look at the line, but only a couple of points here are waiting it out. But in general, so I guess, that I'm not saying ignore the regression line, but in general, there might be sort of if you think of it almost like the bell curve, that if you have more activities, maybe you've gotten to that extreme point where okay, now I'm having sleep problems. Okay, so I think that if you want to make that intrinsically to you, think about having an extremely busy day and how it might be harder potentially to wind down at night. That's kind of how I make sense of these data. Whereas in the depressed individuals, there's a really strong pattern going the other way, right? Showing that the fewer activities are associated with more problems with sleep maintenance, okay? And that the less problems with sleep maintenance, okay, less WASO, is associated with more activities. Okay, I think in the interest of time, I'm going to skip out of that and kind of talk a little bit about light exposure. This was another study that we published a little while ago, but I think it's kind of worth just repeating here because we've talked a lot about light exposure. And what we found in this study is that variability, that SRM index, actually has very strong associations with the amount of daytime light we get, okay, and that light in general has very strong relationship with depression when we control for the amount of habitual behaviors. Okay, so this is what we call a mediation relationship, which is really nice because it suggests two things. It suggests that social rhythm metric not only has to do with light, it really supports that Mistelberger model, but it also suggests that what I find interesting about this model is that even when we control for light, when we do it opposite, we still get that relationship between the SRM and depression. So there's some unique contribution that our behaviors are making above and beyond that of light. Okay. All right, so I want to make sure we have time for questions, so I'm going to kind of move through this. Okay. And I want to talk a little bit about sort of the final, to talk a little bit, I'm going to go back and just look at this model one last time, because I want to say, you do PTSD, where does that fit in here? Okay, well, if you look at this model, PTSD is a really nice model for an extreme stress disorder. And so what we found is that, we've gotten a chance in our research to look at social rhythms, to look at mood regulation. We've looked at sleep, we've looked at some attributional style type things, and we've looked at exposure to light. But where does this event kind of fit in? And so one way we did it is we assessed stressful life events, but we said, well, what about traumatic stress? It's a really nice model for an example, to a certain extent, of a population that has really had exposure to extreme stress and has kind of gotten stuck in that area. They haven't been able to get resilient and move past that, and that kind of fits with the theory of the model. So just to remind you about PTSD, I want to show you that there are 2 symptoms that are embedded within there that could be considered sleep-related, or for sure one that might be considered chronobiologically related. So there's the 3 clusters: intrusion, avoidance, and arousal symptoms. And so we see nightmares and both difficulties falling and staying asleep in there. Okay, what do we know about PTSD and sleep? Well, first of all, just like with depression, we know that PTSD has a lot of problems with sleep, increased problems falling asleep, nightmares. Patients report a lot of problems staying asleep. But I just want to point out that unlike depression, there are some real mixed reports in PTSD with sleep in terms of objectivity. So when we hook patients up into PhD, when we really measure their sleep more objectively, sometimes we don't find differences in sleep. There's one huge study that found no differences between patients with PTSD and patients without on their sleep. Okay. Now, there is a really nice meta-analysis that came out relatively recently, and they found modest differences in patients with PTSD. Generally, we're supporting some sleep continuity disturbances, some problems with REM, especially maintaining REM. One finding that has come out very clear is something called increased auditory awakening thresholds. Yes, it's increased. So patients with PTSD, we hear them say, I can't fall asleep, I'm listening to everything, I'll wake up at the drop of the hat. But the research actually supports the opposite, that there's a harder time awakening patients with PTSD. Okay, and then there is some data showing that there's automatic motor behavior, a lot more of that hitting, or fighting or moving around in sleep. But again, that hasn't been super clear, and they've only looked at it in terms of limb movements, not in terms of other video indices we'd like to see. What about social rhythms? There's only been one cross-sectional, and they're very similar to depression, that patients with PTSD have fewer habitual behaviors, they have fewer consistent routines. There's, to our knowledge, been no assessment of ambient light exposure in anxiety, but there's good reason to think that, because we know that when patients with anxiety disorders, what they like to do is avoid. They like to stay inside. The more you stay inside, the less sort of exposure you'd have to light. So again, we don't have a lot of support for that, speculative at this point, but I think that there's indications out there, and I think it might also lead to one thing that we do see, which is a problem with the dysregulation of the cortisol rhythm, which might indicate that there is some form of circadian rhythm disturbance going on. Okay, so I just want to do a brief summary before we talk about the trial that we've had going on. What we found is that if this has been a lot of data, I just kind of want to summarize that light is the most powerful Zeitgeber. The social and circadian rhythms co-vary. There's good support for the social model in depression, but we definitely need more for research in anxiety and PTSD. And the cool thing about this model is it really implies that a change in social rhythms, and this is why I love this area, can really lead to potentially biological change, even if it's via exposure to light. That's pretty cool. Okay, that just behavior change alone can change our biology. Okay, so how does this all have to do with treatment? Well, there's a really cool therapy out there called interpersonal social rhythm therapy, and it's been pretty well tested in bipolar disorder. There's been at least one good randomized clinical trial, and what they found is that when they compared this treatment to intensive case management, they found that IPSRT, acute phase, was better than intensive until remission. Patients with bipolar had a longer survival without a new mood episode, and it correlated with higher social rhythm stability, so that looks like that's a mechanism for change there. They also found that higher social rhythm scores at remission were associated with a reduced likelihood of mood recurrence later. So based on those findings, we said, hey, patients with PTSD have problems with this too, so can we apply something can we apply the same type of treatment to that? So what we've done is develop, and I say develop, it's really an integrative therapy that's been developed by others, but we've pulled together different elements of the interpersonal social rhythm therapy and cognitive behavioral therapy into a treatment we call cognitive behavioral social rhythm therapy. And the goal of this therapy is to increase the consistency of social rhythms, so in effect, help patients develop a routine, to increase their exposure to light, especially morning light, because we know that's important for timing the rhythms, and then also sleep with a major focus on how to recover from life events. Very little in this therapy addresses trauma. In fact, there's no place really in the manualized protocol to go back and talk about trauma. So why do we do this? Well, because we feel like it's important. I've worked in psych settings, I've worked in sleep settings, and you see patients get bounced back and forth a little bit. So to address the psych and the sleep together is really nice for patients, I think. We developed this for a comorbid veteran population that has so many things going on with. 50% of veterans with PTSD also have depression. Felt like we really wanted a group modality because that's how it works in the VA. There's very few resources for many, many patients. So, piloting a group treatment seemed especially important. And the fourth reason, again, we don't expect this therapy to solve PTSD, but was to improve readiness for PTSD exposure therapies. They have a 30% dropout rate. It's pretty high. You get a lot worse in those treatments before you kind of get better. So the idea is if we can help these patients behaviorally self-regulate, then maybe they can go on and do better in PTSD-focused treatments. It's a manualized group therapy, 12 weeks, 2-hour sessions. Okay. I think I've probably given enough of that at this point. Let me kind of get to some of the findings that we've had. So this is our open trial results, and I just want to say we're about halfway through our randomized control trial. But we've looked at 28 veterans in 6 separate waves, 1 wave of women. Okay, it was 4 women vets. The mean age is 53, so a significant number of vets in our cohort were of Vietnam era. To be in the study, they have to have PTSD, and it was moderate, but generally it was severe that we got, as well as moderate depression. Just to give you an idea of the severity of our sample, 36% had had a suicide attempt at some point in the past, and 46% had been psychiatrically hospitalized. So this isn't like an easy group that we're working with. 71% had combat. What other traumas were there? Things like military sexual trauma for both men and women, car accidents, other accidents that occur in the military. They could have childhood abuse, but we asked that it wouldn't be the major trauma that they were focusing on at that time, the major one causing the majority of symptoms. Okay, so when we look at our findings, this is with the 28 veterans, and again, no control group. You can't say that this is, working better than like treatment as usual or anything like that, but it can give us an indication of how well this therapy is doing. And generally, patients started off, it took them about 50 minutes to fall asleep if we look at our sleep outcomes, and then by the end of treatment, they were doing much better with their sleep, and down to the 3-month follow-up with receiving 22, and only taking 22 minutes to fall asleep, which is nice because anything less than 30 minutes is considered not as much of a clinical problem. If we look at the number of awakenings, we found that it went on average from 2.4 a night to about 1.3 a night at 3-month follow-up. And I just want to say DSD stands for the Daily Sleep Diary, which is considered the gold standard in our field for assessing these types of things. Wake time after sleep onset from 50 minutes down to 20. And their total sleep time went from up about 1 hour from baseline to 3-month follow-up. So it's really nice to see these gains. And generally, you can see that there's a bit of a dip from post-treatment to 3-month follow-up, but not too bad. That's kind of what we would expect. On the depression scale, interestingly, I thought we'd have more of an impact, and right at post-treatment we did, but we found that that wasn't as resilient over time. Although, it was still clinically, it's still significant. I don't know if it's clinically significant. It's nice, though, because it modeled exactly what the patients were saying. If nothing else, our ratings are worse than what the patients are saying. The patients are saying they're still better, they're doing well. Not at remission level, though. Just want to point that out. This was the finding that we absolutely did not expect in the least. The CAPS is the kind of gold standard for PTSD. 50 is considered the cutoff for clinical. So we saw a huge drop in PTSD symptoms that maintained 3 months post-treatment. Okay, we didn't expect that at all. We do no form of exposure therapy in this treatment. And I say that, but some people might say getting out of your house is a form of exposure therapy, and we certainly encourage that. Okay. So big drop, and it modeled patients did the same thing. And generally, a 10-point drop on the PTSD checklist is considered very clinically meaningful change. It's a mean. You know, we can't say that every patient had that, but in general, there's significant data to suggest that this is a pretty good treatment. We have something going on here. Maybe. So where are we going right now with our directions in our lab? Go. We want to continue to score our SRM from our open trial. We haven't published because this measure takes so long to score and then run the variability analysis. We have to do patient by patients for 12 weeks of data, and we have daily data. So we really need to get on it, though, just to let you know. Come on. Okay. We need to examine whether it improves response to exposure therapies, because that's one of our thoughts is, hey, maybe this will help out. I do want to say, and I think that this might be why I have the stars, well, not exactly why I have the star, but I do want to say that our attrition rate in this therapy has been really low. It's only been 18%, which is really nice. And then right now in progress, we have a randomized double-blind controlled trial examining cognitive behavioral social rhythm therapy compared to present-centered group therapy, which was a major, it's sort of the major attention control group that they used in all the exposure therapy trials. So it's a nice attention control group to actually see if this works. And I just want to acknowledge it takes a village. No, I just wanted a big village. Now all my talented trainees, especially Monica Kelly, who has pretty much been my right hand throughout all of our studies and kind of developing our studies, and also Dick Bootson, who's here, who's been my mentor since 2000, through my postdoc since 2003, and then our sponsors as well, including the Department of Defense, who's funding the randomized control trial. Right. And I apologize. We have, I think, a couple minutes for questions. I apologize for going over.